Hello, this is Mr. Cade. I'm going to go over the Chapter 8 notes really quick because I'm limited on time with the video here. So this is the first slide. This is slide 12, and you can see I've got two different colored lines here. And this just means that the red line, red underline, and the blue underlines, they are synonyms of one another. So the light dependent reaction, light reaction, those two are synonyms. They mean the same thing. And same thing with the light independent, dark reaction, Calvin cycle, those mean the same thing, okay? The comment that I wanted you guys to make on this is down here, and I'll roll it up just a smidge more. There we go. It's basically going over these vocabulary words. So it's talking about NADPH, that's an energy carrier, and ATP is also an energy carrier. It's just like a battery. Chlorophyll is contained in pigments. You got chloroplasts that are found in the organelles of the cell. And you have glucose, which is the end product of this whole process. Okay, moving on to the next slide, 13. 13 is pretty complex. You got this equation where you got 6 molecules of water plus 6 molecules of carbon dioxide, a little bit of light inside chlorophyll, and hopefully you produce 1 molecule of sugar, which is your C6H12O6, plus some waste, which we enjoy quite a lot, and that's the 6 molecules of oxygen. Now, you do need to add the words reactants, products, glucose, sugar to this equation. I did circle these 3 things because all 3 of these are reactants. Okay, not just H2O and CO2. You have to include the light. Now the comments down here are pretty important. I'm not going to go through every single one of these. Just that the first one here, chlorophyll A, yes, there is chlorophyll B, but chlorophyll A is found in every single organism that photosynthesizes. Okay, every single one, whether it's a little protist or whether it's a big redwood tree. All of them contain chlorophyll A. All right. The stomata, we'll go over that some more. That just allows, that's the port on the underside of a leaf, or on the top side of a leaf if it's aquatic. But make sure that you understand what's going on in this reaction, this formula here. Okay, moving on to the next comment. So this is on page 15. I skipped 14, and it's just a cross section of a chloroplast itself. This is a little bit more detail. This gives you where the stroma is, which is all the space between all of these granum, which are the stacks of green pancakes here, which are called thylakoids. And then these little bridges here are the stroma lamellae, which basically move the sugar water after it's done being processed into sugars. It makes it into sugar water, and that sugar water is then moved out of there into the phloem, which is like a vein system. So kind of like our arteries and veins. With plants, they have xylem and phloem. Xylem carries fresh water. Phloem is sugar water, and it'll take that sugar water back to its fruits. It'll take it back to its stems, its roots, to basically store these sugars. Okay, what I do want you to put in on this for a comment is the stroma equals the space between the granum. Okay, stoma equals space between the granum. And in this space is where you're going to have the exchange of the gases going on. You're going to have oxygen that has been basically produced here and is trying to get rid of it, is in the stroma, and it's trying to get out. You got CO2 that is trying to get in, and you got water that's surrounding everything. Okay? The next slide is a little more complex. This one's got all kinds of stuff going on. And the very first part of this talks about thylakoids, and then a stack of those are called grana. And I've been over that a few times, but I have not been over photosystems. And the photosystems, if I click on my enlarge tool there, you can see I have photosystems 1, photosystems 2, but photosystem 2 comes first. Okay? When they were discovering these photosystems, they first discovered this one, hence the name photosystem 1, but they made the mistake of saying photosystem 1 because some other scientists came along and discovered photosystem 2, and then some other scientists came along and said, wait a minute, 2 doesn't come after 1, 2 actually comes before 1. So they were stuck with the names, and hence it's been that way ever since. So here's how the whole thing works. You've got light that hits this P680. Now P680 and P700, the P stands for pigment. 680 and 700 are wavelengths of light. So you're talking more in the orange to red area here. And this pigment is also going to contain minerals like zinc and so forth, that when UV light hits it, it super energizes those valence electrons. They're already moving really fast. Well, now they move faster, and they move so fast that they basically can't stay there anymore. They literally fly off. And so a couple of these electrons fly off, and they get so energized. That's what this is supposed to be depicting, is they're getting higher, higher, higher in energy level, that they can't be used in P700 to get P700 up and running. They have to be lowered in energy level. And how does the chloroplast actually do that? Well, in the chloroplast, in these thylakoids, it will run these electrons through these protein channels, and it literally pushes them through these protein channels, where it will push them up and then over and then over and then over and then back and forth. Think of it as a hot potato, and every time you pass the hot potato, it keeps getting cooler and cooler and cooler. And eventually this hot potato will get down to the right temperature where you can eat it. The same thing with these electrons, is they're super, super energized up here. They go through these protein channels, and it keeps peeling off just a little bit of energy, just a little bit of energy, to the point where now the atoms that make up P700's pigment can use them. And it's just enough to make them viable. And so they're able to be used. Sunlight hits them, they release their energy in the same way. Okay, so let me now go down just a smidge so you can get all this in. Great, lost it. Have no idea where it went. Perfect. There it is. Okay, so there it is. Yeah, I hit something and it went. So here you have P700 doing the same thing that P680 did. It has a heightened amount of electrons, and these electrons now are again too energized to do much. So it goes back and forth through these protein channels again, taking off just a little bit of energy as it does that, down to the point where now NADP plus can pick them up, use them to bond with hydrogen. And that's the end game. It needs to produce that NADP plus, needs to be able to produce that NADPH. And it needs those electrons to bond them together. All right. Now, it is kind of ingenious because it goes through these protein channels here, which is called the electron transport chain. As it goes through the electron transport chain here, I said it's losing energy, but that energy is not totally lost. It's being used, and that energy is being used to create ATP. So there's two major things that are being created here: ATP and NADPH. All right. Now I said that these two electrons here help energize and kickstart P700. And then I started by saying, well, light over here kicks this thing off, and this thing loses electrons. Problem is, if you keep losing electrons, you don't have any more electrons, and you're going to forget this nonsense. I'm not giving you up any more electrons. I don't care how much light is going to hit me. So the plant's got to solve this problem. And the way it solves it's really ingenious. It takes some of this ATP, not all of it, but some of it, and it splits water into its components. So it has O2 and H plus now ions. So you have O and two hydrogens. Okay, so you have one O, two hydrogens. That used to be water. You split that. Now you take these hydrogen ions and you move them over here to the NADP plus so that they can combine with that. And then you take these O's and you allow them to bond together. And now you've got oxygen that is basically not needed there. And so you need to get rid of it. That's where our oxygen comes from, is it literally takes that oxygen that it got from water and moves it out of there. Now it's also released all that energy, because when you break bonds, you're releasing energy, and that energy is being released. That energy is now replacing the electrons that were lost by those atoms that originally lost those electrons due to the light hitting them. Okay, very complex system. Just note what you really need to know out of this is a few things. One, that to kick the whole thing off, water's got to be split into its ions. O2 is basically produced here at this stage. It's produced the very first thing, it's the very first product that's made is O2, and it's gotten rid of in the very beginning of the light dependent cycle. And then you have photosystem 2 that is releasing its electrons, but they're so energized that they now have to run through the electron transport chain, or the ETC. As it runs through the ETC, that energy is being used to create more ATP. And now those electrons are de-energized just enough to where they can be used to replenish the electrons that are lost here in P700. And then those electrons are then passed on to help combine or bond NADP plus with hydrogen. Okay, and I know this is a little outside, and this thing is a little wider than my screen. So this whole system wants to go for the end game of producing two NADPHs, ATP, splitting water into O2 and hydrogens, and those hydrogens are then used and passed over to the NADPH. So let me put this down. So what do you need on this? I would outline and draw a box around this. I would draw a circle around here and draw an arrow over to here. Again, to do that, use the scribble tool. That's the line tool over here, all the way down the bottom. They call it the scribble tool. Okay, and I'm going to end this video here, and I will continue on with part 2, because I'm at almost my maximum time for this one. And I will pick it up and explain the rest of the comments that I've made to these slides here on part 2. All right, so I'll end it here.